we're going to look at cell membrane. So before Easter, we were sort of looking at the different parts of the cell and all the different organelles that were involved. Once you basically get into A level, we then sort of spend a lot of time on its own on the cell membrane, and that's because it's quite complex and there's actually a lot more to it than we initially sort of say. So we're going to look at cell membrane, the different layers of the cell membrane, some of the theories behind the cell membrane, and then possibly, if we've got time, have a look at different transports across the cell membrane. If not, we'll then do that tomorrow. So some of that will be a recap, some of that will be slightly new things. But yeah, so this is sort of our view of a cell now at this point with all the organelles that we should now know at A level. So we've got our nucleus, nucleolus, our endoplasmic reticulum, Golgi body, centrosomes, mitochondrion, microtubules. We have these things called secretory vesicles, which I won't have really mentioned too much yet, but we will look into them in a moment when we go under cell membrane a bit more. Cell membrane, lysosomes, cytoplasm, and ribosomes. So what we're going to do is we're going to look at cell membrane in a lot of detail. So for gradings, C grade is understanding why cell membranes are important. B grades, we're going to explain why phospholipids form me cells in water. So actually we've spoken a bit about phospholipids before when we had that whole lesson on lipids. So we're going to look at that in a bit more detail, and because there's lots about lipids in with the cell membrane. And then A is to be able to describe the fluid mosaic model of a membrane structure. So what's actually quite good is to sort of start here with diffusion, because actually diffusion happens a lot in and out of the cell. So obviously diffusion is the movement of particles into and out of the cell across the cell membrane. So this is now our precise A-level definition for diffusion. It's quite good to learn this. I'm just going to focus on some of the keywords. I'm going to read it to you and then pick out some of the keywords to really focus on here. So diffusion is a net passive movement of particles from a region of high concentration to regions of lower concentration, and it continues until the concentration of substances is uniform throughout. So first bit that we've focused on is net passive movement. So net movement is the overall general movement from area of high to low. Why we use the term net is because actually some of the particles will actually stay where they are, and we'll still keep some in this high area. We sort of get this warped view of it sometimes when we're doing GCSE, but actually, let me just annotate on this. We get a bit of a warped view, but it ends up looking like this. And so if we had high concentration over here and low, and then we've got time passing, and so then this would be high concentration and low. We sort of get this view that we've got particle, 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 loads of them over here, maybe one over here and low. And they're going to move from high to low, which is true. And then it ends up looking like this. Maybe we've got one there, and we've got loads where it was low, which has now become high. And that's not really what happens. And so when we talk about this net movement, it's actually that if we have a whole room, we've actually got the region where we've maybe let out this, let's say it's a perfume bottle, for example, the particles is high to begin with, then the whole rest of the room is low. So thinking about Brownian motion, thinking about back to how gases and particles move in physics, they're actually just going to move out. So if we call this over time, I've only got 4 particles there, spread out quite evenly between each other. So it's a net passive movement. Passive meaning does not require any energy, it just happens by itself, of particles from region of high concentration to regions of lower concentration. And that's really important, this plural and regions. So we're not actually saying it's going high to low like this. It's actually, this is a high area, the rest of this area is low, so these particles could sort of go anywhere in the low region. And this continues until the concentration of substances is uniform throughout. And so what we mean by that is basically, if we show this in a different way. I'm going to use the example of food colouring in water. So got a beaker and we've got water in this beaker. I'm going to draw blue to show that it's water, but I'm going to just put some food colouring in. So I've got water in there, I put some food colouring in, I drop it in here. Over time, this water will diffuse from this high region in the middle out to all the other regions. And when we talk about this uniform concentration, what we mean by that is that we end up with something that looks like this. It's red throughout, so it's that equilibrium. It's like going back to that idea of equilibrium at A level, at chemistry even, that everything's spread out, it gets to that area of equilibrium. So it continues until the concentration of substances is uniform throughout, so we've got some uniformity between it. They're all very equally spread out, so that's really quite important, and those extra little bits now at A level to remember for diffusion. So let's go back to mouse, let's close that, let's carry on. So Fick's law. Fick's law is something actually that we're going to look at in a bit later, but I'm going to just talk about it now quite quickly. Fick's law is actually something that we'll look into in more detail when we're doing about the lungs, but it is important with cell membrane as well. And you'll sort of see why we're talking about diffusion and Fick's law here in a second. So Fick's law is the rate of diffusion is proportional to both the surface area and concentration difference, and is inversely proportional to the thickness of the membrane. So basically, what we're saying there is the rate of diffusion is proportional to surface area and concentration difference. So the larger the surface area, the larger that concentration difference, the rate of diffusion is proportional to it, so it's going to increase. The larger the surface area is, the larger the concentration difference is, the faster that rate of diffusion is, and is inversely proportional to the thickness of the membrane. So basically, the thicker the membrane, the slower the rate of diffusion is going to be. So we want, basically, the key premise here is saying the larger the surface area, the higher the concentration difference, concentration gradient, and the thinner the membrane, that is going to give us the fastest rate of diffusion. And so that's quite good and important to sort of keep in our minds and remember when we're then going to go and look at cell membranes in a moment. A little bit of extra information. Cystic fibrosis comes up sometimes in the questions on A level. And cystic fibrosis is a hereditary issue that affects the lungs. It also affects the pancreas, and it also affects the reproductive organs. And what it is, it's basically this production of an excess of mucus. So if you think about Fick's law, if cystic fibrosis is increasing the amount of mucus that's in your lungs, it increases the diffusion distance because you've got extra mucus in your lungs. So it's actually longer, takes longer for the oxygen to diffuse from inside your lungs into the bloodstream. But it also reduces surface area for gas exchange because the mucus will be blocking certain areas as well. So cystic fibrosis causes diffusion distance to increase and reduces surface area. So we've got, so basically it increases the thickness of the membrane. Diffusion distance and thickness of the membrane here is the same sort of thing. Mucus acts as thickening the membrane. And then, yes, we're also going to reduce the surface area for gas exchange there as well. So cell membranes, cell surface membranes, here's an image of one. Membranes are found around all of our organelles. So actually, we have a cell surface membrane around the outside of our cell, but we actually do have membranes around all of our organelles as well. So let me skip this bit a second, we don't want that. And so we've got an image here. Let me get rid of those annotations. We don't want them. It's an electron microscope image of cell membrane. So this would be, the green section is inside the cell, blue is outside the cell. And so then the blue line with like an orange image in between, that is our cell membrane. When we're quite used to drawing cell membranes back at GCSE, we usually just draw a cell membrane as a singular line. And if it's a plant, we then have our singular line for a cell membrane and another line for our cell wall around the outside. But actually, this looks more similar to a plant cell wall and cell membrane, but it's not. It is our cell membrane. Actually, our cell membrane is a double layer. It's a bilayer. So this is one layer of it. Oops. But one layer of it and then another layer of it on top of each other. So that's what we're going to look into now, is actually what is the bilayer of the cell membrane. That's really important to remember. So I'm going to look at different parts of the cell membrane. So the cell membrane is made up of phospholipids. If you want to at some point, you could always screenshot this page and then fill this in as you go. So we're going to look at the different parts of the cell membrane and then micelles, fluid mosaic model. So phospholipids, we've talked about phospholipids before, and we've talked about fatty acids. So fatty acids, I'm going to draw you a fatty acid now on here. A fatty acid, we have got our glycerol, and we have our three fatty, our glycerol and our three fatty acid tails. So if we drew a lipid, for example, that is what a lipid is. So that's our lipid here. A phospholipid is very similar, but we've removed one of those fatty acid chains and added a phosphate head onto the glycerol. So this is our phospholipid. You remember this from before. It's made up of two fatty acids attached to a glycerol, and on the glycerol we also have an inorganic phosphate attached to that. So phospholipids, you probably can recall this as well, but we'll go through this again, have a negatively charged phosphate group. So the phosphate group up here, let's change the colour so we can actually see what I'm going to write. A phosphate group has a negative charge. And this negative charged phosphate group replaces that third fatty acid. Because the phosphate group is negatively charged, we say the head is negatively charged, and we actually then say that the tail is more positively charged. We're going to go into hydrophobic and hydrophilic in a second, so there's some words that we maybe want to try and recall and remember. Phospholipids. So phospholipids, these are the most abundant molecule within the plasma membrane. So we often say plasma membrane, cell membrane. We sometimes call it plasma membrane. We sometimes call it a cell membrane, cell surface membrane. Again, it's one where we have a few different words each time. It's actually made up of quite a few different molecules, but our most abundant molecule that we find within the plasma membrane are phospholipids. And they're the key structural part of a plasma membrane. And the nature of these phospholipids is the key to the function of a membrane. We say it's a bilayer. So we say there's two layers, and actually that really applies to that hydrophobic and hydrophilic part of it I was talking about, and the positive and the negative ends. So why is this not working? Okay. I'm going to draw. There we go. There's a phospholipid. So there's the same image from phospholipid before. Its head. So we refer to it as having a head and a tail. The head is made up of the glycerol and the phosphate, and we say the tail is made up of fatty acids. And it does look a little bit like a head and a tail. So we say the head is hydrophilic. So it's negatively charged. It's hydrophilic and therefore likes water, whereas the tail is hydrophobic and therefore dislikes water. You are allowed to use the terms likes and dislikes with water. You can say it's more attracted to water, it repels water, but anything along those lines is hydrophilic and hydrophobic. Why this happens is because the head is polar. So The head is actually combined of a positive and a negative charge, where the tail is non-polar, so it has no charge at all. So if you think back to that last session we did in chemistry about polar molecules, we end up here with actually different polarities in different sections of a molecule. So actually the head is polar, it has a positive and negative charge. The phosphate is slightly negative, the glycerol is slightly positive, and then the fatty acid tail is a non-polar part of the molecule, and so this has no overall charge. So the role of our plasma membrane. So the point of our plasma membrane is to allow different substances in and out of the cell. Again, I'm going to show you an image like this because it's always really important to remember that cells are a 3D structure and they're not just that flat sort of circle that we look down in. That's as if we've got one, cut it in half and just looking straight down in it. And so these images are sometimes quite helpful, and you get lots of different ones like this in different textbooks. So the role of the plasma membrane. The point of it is that it separates the cell contents from the external environment. So all of this stuff inside your nucleus, your Golgi, your mitochondria, your chromatin, your vesicles, they are kept inside and out of everything outside the cell. So it's basically a protection, and it's a capsule. It keeps everything there, so it contains everything. So it separates our cell contents from the external environment, but it is also able to control the exchange of substances between the cell and the external environment. So we're separating what we've got in there from the outside, but it's not a barrier that's impenetrable. Some things can get in and some things can get out, but it actually can control them. So it has a control element to it. So it's not just anything coming in, anything going out, there's a control. And to help with that, it contains receptors for cell recognition as well. So this is how our cells interact. So if we go back to basic year 7 biology, we would say groups of cells together would make tissue. And these are cells that work together, doing a similar job. Well, actually, the cell needs to be able to recognize that there's another cell nearby, or the cell needs to recognize that there's this protein nearby, or the cell needs to recognize there's a hormone coming in. So actually on our plasma membrane, we also have receptors for cell recognition, working out what's going on externally around it and if anything needs to change. So here is that image of the plasma membrane again that we had at the beginning. Now it is 7 nanometers diameter. We're not doing a practical. Actually, I'm going to talk through the practical really quickly with you, actually, because it's something that you can just quite easily do at home. So I'm saying you can get a Petri dish, you can get a glass, anything that you could see through, and a spoon, and you put water in it, and then you put some oil in, and you look at what happens. And it's that idea of the separation, that the two won't mix together. And if you stirred it with a spoon, does it make any different shape? Try different types of oils. Is there any difference? And it's thinking about why won't the oil and water mix? Well, oil, as we know, is a lipid. And if we're thinking about that, thinking about the hydrophobic, hydrophilic parts of it, we'll have some sort of similarity there. So this is what our bilayer looks like. This is what our phospholipid bilayer cell membrane really looks like. So we've got loads and loads of different parts. So we say that the phospholipids are the most important part, they're the main bulk of it. And what we do is we work as a bilayer. So because the heads are hydrophilic and they like water, basically what happens is they all line up next to each other, and we've got this top layer of the bilayer, and they're all touching water. The inside of our cell, as we know, 90% of our body is composed of water. Inside of our cell's got a heck of a lot of water in too. So the heads are on the inside, and then we've got this 7 nanometer gap between them that's actually just made up of the tails, and the tails attract to each other because they're hydrophobic. So they attach to each other, they're attracted to each other, and the heads are on the outside. So that's our phospholipid bilayer, and that's what we mean by that. It's one layer of phospholipids on top of another layer of phospholipids, tail to tail. And we have actually lots of other stuff going on inside here. So, and this top section is the outside of our cell. I'm just going to get a pointer so we can see this better. So this is the outside of our cell. So we have extracellular fluid out there. So it can be water, it can be anything that's in there. In between the phospholipids that make up our bilayer, make up the membrane, we have cholesterol. And we're going to talk about what cholesterol does to the membrane in a bit. But cholesterol plays a really, really important part on the functioning and the fluidity of our membrane. So our membrane actually is fluid, it's able to move. And cholesterol helps control and regulate how fluid our membrane is. And so we've got cholesterol in there. We also have these really, really big things that are being shown in blue. So for example, we've got this one over here. This is a protein, this is a transport protein. So this is one of the ways that some molecules can get in and out of the cell. Some can actually get through down past the bilayer. Some are too big, so some need to come in through this way. Here we've got a binding site. So this is where maybe a hormone might attach and then something else happens somewhere else. So this is like a receptor. So this is an area actually where something comes and binds onto the protein. This is a receptor protein. It binds at the binding site, so it could be a hormone that's then going to trigger some other reaction inside the cell. So that hormone doesn't need to enter the cell, but it needs to get to the cell, bind to it, and then another reaction occurs somewhere else inside the cell. And then here we've got a recognition protein, works in a similar way, but we have these carbohydrate groups hanging off the side and they work a little bit differently. Then inside we've got protein filaments. They sort of work as an extra sort of support, and then we also have glycoproteins on the outside. So it's actually really important to remember that we have all these different parts of the cell membrane, and it's not just this phospholipid bilayer. We've got the phospholipid bilayer, cholesterol in between the phospholipids, recognition proteins, receptor proteins, and transport proteins, and glycoproteins on the top as well. Pass this about 5 times. Okay. 1 second. What have I done? Okay, so when our phospholipids are exposed to water, the tails will always move away from it, and the heads will move towards it like this. So here is an image. If it will open, why is this not working? Why is everything frozen? One second. Okay, so when we think back to that image, I was saying that we've got the phospholipid bilayer, and then obviously we've got cholesterol in between it. So we're going to look at what the role of the cholesterol is and why it's there. So cholesterol is actually another type of lipid, and it has the molecular formula C27H46O. Cholesterol is really, really important in controlling membrane fluidity. So when I said membranes can move, what the idea is that actually the outside of the cell, there's water, there's fluid. Inside of the cell, there's fluid. And the heads, so these hydrophilic heads, are attracted to the water. So as the water moves, these heads will move so that actually they have a fluid motion. So actually the membranes can actually move apart, move together. It's not a rigid structure, it can move. So cholesterol actually reduces the fluidity. So cholesterol comes in and it basically almost acts as a brick, an anchoring point. So the more cholesterol is present, the less fluid the membrane is. Therefore, it's less permeable. Now, we want some cholesterol in there. It's not that cholesterol is really bad and we don't want it there at all. We actually do want some cholesterol in there because we wouldn't want it too fluid. If it's too fluid, we could have all sorts coming in and out and there's less control over it. So we want the right amount of cholesterol. So we want some in there to reduce the fluidity. However, if we have too much cholesterol in there, that will reduce the amount of different substances that can actually get through, permeate through the membrane. So it's a very fine balancing act. So cholesterol is also important in keeping our membrane stable at normal body temperature. So without it, cells would burst open. So our normal body temperature is 37.5 degrees. Cholesterol is really great at actually keeping our cells intact. It's another anchoring point that actually helps keep them intact at our temperature of our body. If you think 37.5 degrees is actually quite warm, the amount of energy that we're providing to all the different molecules, they'd move around a lot and they'd burst open. So for cholesterol, again, it works as an anchoring point to keep everything there. Again, too much cholesterol, it's not going to be fluid enough. Too little cholesterol, too fluid. So it's a real fine balancing game with cholesterol in our cell membranes. We also have proteins in our cell membranes, like we talked about before. So we have transporter proteins and receptor proteins. So proteins typically make up 45% of the mass of a cell membrane. This can vary from 25% to 75% depending on the cell type. So some cells will have a very small amount of proteins there because maybe they don't need much coming in. They might not need much coming out. They might not need many recognition proteins. Other ones might need loads. So for example, neurons are going to have lots of proteins there because actually they're going to need to take in lots of neurotransmitters, which is going to be recognition proteins that were involved in that. So they would probably be more like towards the 75%. As an example, muscle cells, they're going to be quite high in the amount of protein. Skin cells, you're probably looking at like your medium range. I'm trying to think of something that would have a low amount in. Hair cells probably would have a lower amount. So we can have two different types of protein. We can have integral, also sometimes called intrinsic or transmembrane, means the same thing. It's a protein that goes through the membrane. So this protein spans the whole width of the membrane, that whole 7 nanometers. So that's an integral protein. And we can also have peripheral or extrinsic proteins, and these are confined to the inner or outer surface of the membrane. You can also have peripheral proteins attached to integral proteins as well. So our purple one on there is an integral protein, goes the whole width of both bilayers. Green one is a peripheral or extrinsic protein on the top layer. Yellow one again is another peripheral one. It's on the bottom layer. It's also attached to our integral protein, so they can work together. Also, many proteins are glycoproteins. So these are proteins that have an attached carbohydrate chain to the top of them. So the purple protein there, we will describe it as being an integral protein that has a glycoprotein. So integral proteins, what are they? What do they do? So these are the ones that span the whole width of the membrane. Many integral proteins are carrier molecules or channels. So we call them carrier proteins, channel proteins. And what they do basically is these help transport substances such as ions, sugars, and amino acids that can't diffuse across the membrane but are still vital to a cell's functioning. So some molecules can literally just diffuse through the membrane. So things like oxygen, that can just diffuse straight through the bilayer lipid, phospholipid bilayer of the membrane, and that can just diffuse through these gaps. Ions, sugars, amino acids. Ions are charged, so it's hard for them to get through. Sugars, amino acids, they're also large. They can't diffuse through. So it's a mixture of, depending on whether it's the charge or the size, that stops them from being able to diffuse through. But the cell still needs them. But if we had a gap big enough to let them through, the cell would just fall apart. Or, for example, it's an ion, it can't get through because it's charged. So instead, we have these integral proteins to help us. So the carrier molecules, they literally work as a tunnel, a pathway in and out of the cell. So molecules can leave the cell this way, molecules can enter. So you'll find lots of channel proteins for things like sodium ions, potassium ions, calcium ions. Other integral proteins are receptors for hormones and neurotransmitters, or enzymes for catalyzing reactions. So for example, this one down here, it could be an integral protein for a neurotransmitter. So a neurotransmitter comes and attaches on here, and then something else happens down here. Or a hormone comes and attaches itself here, which then sets off another reaction inside the cell. Or, like we said, an enzyme for catalyzing the reactions. Our peripheral proteins, our proteins on the outside, these may be free on the membrane surface, or they can be bound to an integral protein. So like that yellow one was bound, it can actually also then get released, or it can be free. So peripheral proteins on the extracellular side of the membrane, so this is on the side that's to the outside of the cell, they act as receptors for hormones or neurotransmitters, or they're involved in cell recognition. So 3 different things they can do. Many of these are also glycoproteins, so they have the glycoproteins attached to them, and they have carbohydrates attached to them. So yeah, so they're going to act as a receptor for either a hormone or a neurotransmitter, so it's going to recognize and receive those hormones, those neurotransmitters, and then it sets off a chain reaction of other events inside the cell, so the cell then responds to it. Or they're involved in cell recognition, so recognizing other cells nearby. And many of them, like I said, are glycoproteins. Peripheral proteins on the cytosolic side, so that's the cytoplasm side, so the inner side of the cell, are involved in cell signaling or chemical reactions. So for example, that yellow one there, these can dissociate from the membrane and move into the cytoplasm. So often what will happen is maybe this was also a receptor protein, a hormone comes and attaches on, and then some cell signaling then needs to carry on. So it might be that something's signaled a hormone, or something else is received another piece of information, and then another chemical reaction or another cell signaling happens inside. So it might be the chemical reaction needs to happen because we need to produce something, or it could be cell signaling because it might be actually signaling the cell needs to replicate its DNA and then go through mitosis. It could be that the cell needs to die. So there's all different things. So they can actually dissociate from the membrane and then move further down and into the cytoplasm. So phospholipids. This is the one I was trying to get work earlier. So when phospholipids are exposed to water, the tails will move away from it and the heads will move towards it. And it's frozen again. What is happening here? Integral proteins. If I open this up, are they not going to show again? Not quite sure. Here we go. I must have put some weird timer on. So when phospholipids are exposed to water, the tails will move away from the water. So this is our example. We've got some water here. The heads would sit in the water and the tails will sit out of the water away. So that's what would happen there. They can also form micelles, which looks like this. So this is why they make a bilayer, because we have water on the outside and the inside of the cell. We have the polar heads here, we have the tails there, then we'd have another set of tails and heads. So other than forming a bilayer, they can also form a micelle, and the micelle is when you have all the tails coming in and attaching to each other, and then the heads are on the outside. So now we know the different parts of the cell membrane and micelles and functions. So we're going to look at what the fluid mosaic model of the plasma membrane is. So I've spoken a bit about the plasma membrane being fluid and that it's able to move. So we're going to actually look at the fluid mosaic model, and that's the actual name of the model of the fluidity of the plasma membrane. So what the fluid mosaic model is, it's a model that basically suggests that the membrane is made up of a bilayer of phospholipids that can move around relatively freely, or fluid. So that's sort of we're happy with that understanding so far anyway. That's sort of what we've said. here we go. So it can slightly move about. Phospholipid bilayer, 7 nanometers, 7.5 nanometers wide. So if we have a look at the proteins, so some proteins have hydrophobic and hydrophilic regions. Some are on the inner side of our membrane and some are on the outside, and others across the whole membrane, so there are transmembrane ones. Many are transport proteins that allow substances to pass through their channels into the cell, and some are enzymes. So channel proteins pass through the whole membrane and are involved in diffusion. So a channel protein basically works like a tube. It still works on the effects of diffusion. It's still that net movement. It's still passive, but we've just got this tube to help it pass through. Carrier proteins, these pass through the whole membrane again, but these are involved in active transport, so energy is required for carrier proteins to work, whereas channel proteins just rely on diffusion. So you've heard me mention glycolipids, glycoproteins, so we're going to actually have a look at what they are in a bit more detail. So glycolipids and glycoproteins, these are those ones that have the carbohydrate chain that projects out into the environment. What this is able to do is these can form hydrogen bonds with water to help stabilize the membrane. So when we were talking about hydrogen bonds the other day, hydrogen can form those bonds with oxygen. So then the oxygen in water is what you can form the hydrogen bond with. Some glycoproteins are actually receptors for hormones, and one glycoprotein group called antigens are responsible for cell recognition. So the cell is not attacked by the immune system. So actually that's really an interesting part. So when we talk about antigens, and that comes up a lot recently because of COVID, actually when we talk about antigens on the outside of a cell, and we're like, well, what is this? Or the antigens on the outside of a virus or the outside of a bacteria. They're basically involved in cell recognition. So these are basically what tell the other cells what one is and what one isn't, what like basically names them. It's like a barcode. So we actually have antigens on all of our cells, and on a normal cell, we have an antigen there for cell recognition. So that actually our normal cells in our body are not attacked by the immune system. Our body accepts them as being part of their own. This is where the issue comes in with donor organs, because the antigens won't necessarily on the donor organs match your antigens in the rest of your body. So that's why people have to get donor organs that are as similar to their tissue type as possible, and then take immunosuppressant drugs to basically suppress the immune system from rejecting it and trying to kill them cells off. That's not what I wanted. So phospholipid tail length. The longer phospholipid tail length, the less that will decrease our membrane fluidity. If we increase the temperature, our membrane fluidity increases as well. So it's why when we say all bonds can break when we increase temperature, it's the same thing. Those phospholipids are gaining energy, they can move more, the membrane will become more fluid. At higher temperatures, our cholesterol will decrease, and at lower temperatures, our cholesterol will cause our membrane fluidity to increase. Saturated fatty acid content decreases our membrane fluidity. And who discovered the fluid mosaic model? So we're going to have a little look at this part. So the idea of a phospholipid bilayer was first produced by somebody called Jonathan Singer and Garth Nicholson, and these were biochemists from California in 1972. Originally, it was thought that the membrane was a three-layer protein lipid sandwich, and this idea didn't take into consideration basically the fact of the hydrophilic heads of phospholipids tend to be in contact with the water. Once we had this idea of phospholipid heads being in contact with the water, the bilayer model was then accepted based on the evidence from the electron microscope, so like that image I showed you earlier. So then what happened was later experiments then focused on the protein parts of of the membrane. And what happened was scientists found that some proteins could actually be easily dissociated from the membrane by increasing the ionic strength of the solution therein, whilst other proteins couldn't be dissociated without the addition of detergents. So what this did was this actually suggested that some proteins existed on one side of a membrane, whereas others are embedded inside or across the whole membrane. So actually, increasing the ionic strength of solution dissociated some, but not all of them. So actually, it was that thought that actually we've got some on the outside, we've got some on the inside, and we've got some in the middle. So that's how we got that idea of this bilayer. So then they went through something called freeze fracture, and this is a way to evidence this bilayer. So they've said, okay, there's a bilayer. Someone's come up with this idea, great, it's a bilayer. Someone said, prove it. Okay, well, when I add ionic solutions, different ionic strength solutions, some proteins easily dissociate. Others, I can't get rid of them. They won't dissociate. I'd have to add detergents to them. I'd have to break down the cell. So then we're saying, okay. And someone says, yeah, that's fair enough. That's some evidence, but can you prove it to me a bit more? So then they came into this freeze fracturing. So freeze fracturing, this was additional evidence for actually that proteins span the whole membrane. And so those transmembrane proteins, and this came from freeze fracture of membranes. So in this, what we did, basically the membrane's frozen and fractured along the weak points. So they freeze a cell, and what happens is along the weak points between the two layers of phospholipids, so between the two layers of the tails, that's the weakest point, we fracture it there, and then we can look at the two layers. The layers are coated in heavy metal, and then looked at using a scanning electron microscope. Obviously, a scanning electron microscope works in the way that they are hit with a huge amount of array of electrons that hit onto the specimen, which has metal on it, often gold for some different objects as well. And then depending on the frequencies, the different electrons will bounce off it and create different light and dark areas. So the more dense an object, the darker the image. So this would be the results. And so this is the inside of the extracellular layer and the inside of the cytoplasmic layer. So the inside of the cytoplasmic layer, when we were having membranes that went the whole way through, some of them would stick to the cytoplasmic layer more than they would stick to the extracellular layer. So we could see those differences. Actually, we could see that there was all those different amounts of protein on both. So the only other thing I want to quickly show you, let me pause this. I'm recording. Should be screen sharing this. Let me stop my screen share in case I'm not showing the right thing. Let me share it again. Share screen. Here we go. Okay, so we're going to recall that molecules and ions are moved across our cell membrane by diffusion, osmosis, active transport, and then the two new ones we're going to look at is exocytosis and endocytosis. So we talked about diffusion earlier. The difference between diffusion and osmosis, thinking about that, what's the difference between diffusion, what's the difference between osmosis, and the definition of each. So our definitions of each from GCSE is diffusion is, osmosis is our passive net movement. Again, it's that net movement, that overall movement, passive meaning requiring no energy, of water molecules from an area of higher water concentration or dilute solution, depending which way you're going to describe it, to an area of low water concentration or a concentrated solution, through a partially permeable membrane. And that part was always really important. It's a partially permeable membrane, meaning it's a membrane that lets some things through, doesn't let everything through. Diffusion, the passive net movement of particles from an area of high concentration to an area of low concentration until equilibrium is reached. And that's that really important part as well, that we reach equilibrium. Because actually that image in the middle, well, technically, yeah, diffusion's happened, but it hasn't fully happened until we reach that level of equilibrium. So active transport, I'm going to have a look at that in a bit more detail as well. So diffusion, active transport, we know is very similar to diffusion and osmosis, but it's the opposite. So we're going this time from a low concentration to a high concentration, and it requires energy. So that happens a lot in plants through root hair cells, getting different mineral ions in. So we've got an image of a cell membrane. We've got different ways of different molecules getting in and out. So we've got water molecules and sugar molecules. So one way that water can get in is through diffusion. Diffusion is passive and it requires no energy. So sugar can get in that way, water could get in that way. Another way is osmosis, which is your normal way of water to move. And that's going to go through a pore. We'll talk about why the water goes through a pore. But if you think about the hydrophobic and hydrophilic parts of a phospholipid bilayer, why it might need a pore to go through to get in. Active transport, so that requires energy. So that's the removal of, we've got the movement of sugar out of the cell, for example, here, and this is our concentration gradient. So passive and active. We also have another type called facilitated diffusion, which we'll look at in a moment. So diffusion, at A level, it's often referred to just as passive transport as well, so don't let that confuse you if you see that anywhere. So diffusion at A level, we say molecules or ions, we can still say particles, but often in biology we start to say molecules or ions, move down a concentration gradient until equilibrium is reached. Lipid molecules in this image are shown as orange, oxygen, carbon dioxide, polar, so it allows rapid diffusion. So basically, diffusion is a net movement of particles from a region of high concentration to a region of low concentration. And that key word again is net. Net meaning particles move in all directions. Net is that overall movement. And I think about Brownian motion. Obviously, we're not always talking about gases, but it's that idea that it moves around randomly. And this continues until equilibrium. So that is diffusion. Osmosis is our net movement, that overall movement again, of water molecules down a concentration gradient across a semi-permeable membrane. So this is obviously net movement of water from a solution with a lower concentration of solute to a solution with a higher concentration of solute through a partially permeable membrane or semi-permeable membrane. It doesn't matter which way. So again, key points, it's the random movement of molecules. Solute molecules have formed hydrogen bonds and movement is therefore restricted due to their size. So this is how they can move through there. So it's why it's the movement of the water and it's not the movement of the solute. Then we have active transport. Active transport, minerals, some sugars, amino acids, they'll actually move against the concentration gradient. They'll regularly be going from where there's not many to an area where there's lots. So they'll actually move against the concentration gradient. So active transport across a cell membrane requires a specific carrier protein because it needs energy. So this is always going to involve a carrier protein. And it is a movement against the concentration gradient. And this requires an energy input from respiration. So it's that ATP that we talked about a couple of weeks ago. It's that ATP that will be needed. The ATP, adenosine triose phosphate, energy transfer molecule, actually goes in and it changes the shape of those carrier proteins. We call it a conformational change. It changes the shape of those carrier proteins to allow that molecule to leave from an area where there's not many of them to an area where there's more of them. So then I said, oh, there's another type of diffusion, and that is facilitated diffusion. I'm actually going to just hang about actually on active transport for a minute. There's just a bit I want to add to this. I've just lost my pen. So what happens is it's movement from low to high. It requires energy. We've got our specific carrier proteins. What actually happens is when this ATP arrives and we use it, let me just check time. Yeah. What actually happens is ATP turns up. it's used as energy, and what then happens is it becomes, we go from ATP and then it becomes ADP plus PI, which is an inorganic phosphate. So you'll probably remember this from when we talked about ATP before. So what happens is one phosphate is removed by hydrolysis. So what happens is, because one phosphate is removed by hydrolysis, our phosphate becomes hydrated. Lots of energy is released when bonds are formed between the phosphate and the water. Energy changes the shape of the carrier protein. Sometimes, for some reason, we call these pumps instead of proteins. So proteins or pumps. Again, it's just a terminology thing. It means the same thing. So ATP is adenosine triphosphate and ADP is adenosine diphosphate. And then I will talk through facilitated diffusion. Oh, pen stopping me from doing that. And then I'll leave exocytosis and endocytosis for tomorrow. So facilitated diffusion. It's a bit weird. So sometimes molecules can't pass across the membrane by themselves, but we don't need energy. We then have facilitated diffusion. It's like an in-between diffusion and active transport. This is just where we use a channel protein. So it's a molecule that can't just get through the membrane itself. So for example, an ion. So we use a channel protein for the molecule to pass through. So for example, hydrophilic molecules larger than carbon dioxide cannot diffuse through past the hydrophobic tails of phospholipids. Okay, so because of the hydrophilic and hydrophobic parts of it, CO2 can get through fine. Anything bigger than that can't diffuse through, so we'd have to use a channel protein. Anything that is insoluble in lipids cannot pass through the membrane because the membrane is made up of lipids, therefore needs to go into the cell using, or out of the cell using, a channel protein. And molecules that are too large to pass through the phospholipid bilayer will also use a channel protein. Here we go. Here's the channel protein, and this is all that happens. It just moves them in to there, and it can rotate them around. They are specific to the molecule or ion, so it's not a one-size-fits-all. It's very similar to how enzymes work. It's got a specific binding site, and a specific molecule is allowed in. Now, you do get some where you might have sodium and potassium ion pumps, and they're actually for active transport, but it's the idea that you can actually have double ones, but they are very specific. It isn't, oh, we've got a channel protein that lets through everything under the sun. It's very, very specific. We can also have gated ones. These allow for channels to be open and closed. So hormonal change in potential difference to be built up. So again, that doesn't require energy, but it's gated until we've got either electrical chemical gradient, or we've got different potential changes, and then it can open up to allow things through. So summary of passive and active transport. This is a really good way to remember it. So straight through the membrane, diffusion and osmosis, and they are passive. Using a channel protein, facilitated diffusion, also passive. Using a pump, different types, carrier proteins, active transport requires ATP. ATP becomes ADP plus PI. This is quite a good question actually too. I'm just going to leave this here and I want you to have a little look at this, and we'll start tomorrow's lesson on this. This is a 4-mark question from an exam paper about picking out the correct terminology. So some molecules move across the cell surface membrane by passing down a concentration gradient through the phospholipid bilayer. Movement of some polar molecules across the membrane involves carrier and channel something molecules. When this movement occurs down a concentration gradient, this process is called, and when it occurs against a concentration gradient, this process is called. Energy in form of is used in the movement of molecules against a concentration gradient. So whilst this is quite an easy question, I've included it because it's actually showing how important it is that we know our terminology and we know those keywords that we're going to be called upon. So what we shall do is we'll